Welcome to our first webinar of the new year, a beginner's guide to RNA-Seq. I'm John Cafferty, Marketing Manager here at Novagene Europe. Before I hand you over to your host for today, I'd like to thank you all for joining us on this webinar. I'm delighted to say this has proved to be a very popular topic. We had to upgrade our webinar license to accommodate you all. A bit of housekeeping before we start. The webinar runs for an hour and there will be around 15 to 20 minutes for question and answers at the end. Please submit your questions via the Q&A button you'll see at the bottom of your screen. You'll notice that you now have the ability to upvote questions that you like by clicking the thumbs up symbol next to the question. We will try and prioritize the most popular questions, but if we don't have time to answer them, we'll get back to you by email after the webinar. Talking of which, we will share the slide deck and a recording of the webinar via email. You can expect this on Friday. So without further ado, I would like to introduce you to our host for the day. One of our UK account managers is Shama Khan. So Shama completed her BSc degree in cell biology from the University of East Anglia and specifically enjoyed learning about signaling pathways and obviously genetics. After she obtained her degree, she landed her first role within the pharmaceutical and device industry, specifically working with clinical commissioning groups, relaying clinical trial data on novel drugs. During her time in the pharma industry, she completed a master's in medicinal chemistry, looking specifically at drug-to-drug interaction at cellular and genetic level. So she's worked for Novagene since 2018. Shama, over to you. You need to come off mute first, though.
Sorry about that, everyone. So yeah, hello, I hope you're all well. I'd like to firstly say, I hope you're keeping well and safe in these unprecedented times. Like many of you here today, I actually have a little girl as well, and I put her in a crib. So if you do hear anything, I apologise in advance. So I guess we're all in this together. As John mentioned, I would like to introduce myself as Shama, and I live in a place called Southampton, which is south of the UK. I am going to go through a brief overview of our company with the services before I hand over to Joseph Yeo, one of the technical support specialists, who will then go through today's topic, which is a beginner's guide to RNA-Seq. So, obviously, next generation sequencing has recently gained a lot of popularity, especially during this pandemic time, and it is a fantastic tool of research area. At Novagene, we have expertise in a variety of different areas such as biopharma, infectious disease, consumer. Not only do we have a lot of experience, but we're also a global presence around the world. And we have labs obviously in Hong Kong and in America, and more relevant, I guess, to you guys, we have 2 labs, one in Amsterdam and one in the UK. Our UK lab is our main facility in Europe. We opened this state-of-the-art facility back in 2011, and we have 3 NovaSeq platforms there and a wealth of experienced NGS staff. We do have also a number of different services that we can offer, such as whole genome sequencing, RNA sequencing, metagenomics, and also lane sequencing. So in specific, what type of services do we offer? We have experience sequencing viruses, bacteria, plants, right through to animals such as humans. We have decided to kind of like characterize those into 4 main areas. The first is obviously DNA services, which will include human genome sequencing, whole genome sequencing, whole exome sequencing. We also do plant whole genome sequencing as well as microbial community as well. The second, which Joseph will be discussing later, is obviously transcriptomic services. And I'm going to leave that because Joseph will speak to you about that. Then we also have epigenetic services, such as whole genome bisulfate, chip, and RAP sequencing. And the fourth is quite bespoke, and that's to you. So if there is anything that is not in these categories, then we can actually help you with those, such as pre-made libraries and customized analysis as well. So in specific, why would you want to use Novogene? Now, I could give you 1,000 reasons why, but I'm not going to bore you. So I'm going to stick to two main reasons where I think is actually relevant to you guys. The first is technology. We only use the gold standard products, and that's why we've partnershiped with Illumina and use their latest platform, specifically the NovaSeq 6000. The second is quality. We pride ourselves on doing the best quality data for you and ensuring excellent quality. And the way we do that is through quality control checks. We have three main quality control checks. One is when you first send the samples, we can check them and we can check them to see if they are good samples. And Joseph's going to go through that in more details. And the second is once we've actually gone through that using qubit for DNA degradation via gel, but also RNA looking the amount of purity in binalyzer, then we can go and follow it after through to sequencing. We also check after sequencing and also after analysis. Like I said, Novogene pride themselves on providing excellent data, so much so that we've actually got 32 NGS related patents. We've been published in over 580 plus prestigious research papers such as Nature, with a total impact factor of over 4,000. Not only do we pride ourselves on quality, and something that Novogene are very good at as well, is that we also invest a lot in people like myself, an account manager, and we want to ensure that we provide you with the best help and guidance to meet your research goals in the best way possible. We have local account managers in each of your countries that reside in your countries. So when we can actually meet you face to face. Otherwise, we can speak to you on Zoom, or we'll get back to you in a very timely fashion. And here we are in all of our glory. So I'm going to keep this open for a little bit because I know there's a lot of you here today. Please look for your local representative, your local account manager. They'll be so happy if you just wanted to send a message to say hi, I'm looking for some information, or just to introduce yourselves. So that is me done. I'm now going to pass you on to Joseph, who, like I mentioned, is the technical specialist. Joseph's interest in biology stems from his childhood days living in a mosquito-infested country, and he continues to be bitten by other blood-sucking insects, even into adulthood. He studied his Bachelor of Science at Queen Mary University of London. He went on to do a PhD on mobile genetic elements in mosquitoes at the same university. After that, Joseph took on a postdoc position at the University of Glasgow. He joined us at Novogene as a technical sports support specialist in Novogene back in February 2020, in those happy days where we were allowed to actually see each other. And he was there for a full month before lockdown, and he remembers those days with warm memories. So I'm going to now pass you on to Joseph.
Thank you, Thelma, for the introduction. And thank you very much for joining us here today. So I'm Joseph, and I'm part of the technical support team in Novogene Europe. And I will be presenting to you this talk titled A Basic Guide to RNA Sequencing. Now, in this talk, I will cover 4 different areas. So I'll just start off with choosing the right RNA sequencing for your project, and then move on to the Novogene service workflow. Next, I will cover setting up your bioinformatics analysis, before finally talking about the results overview. So on to the first topic, choosing the right RNA sequencing. So you will need to consider a few things for your projects. So to start off with, which type of RNA are you studying? So which type of RNA are you interested in? So as we know, there are different types of RNAs in eukaryotic total RNA. So this from coding to non-coding RNAs. So this is just a graphical representation of RNA by mass. And as you can see, in total RNA, we have the ribosomal, the transfer, and messenger RNA. And then we also have the non-coding RNA, which includes the long non-coding, circular, and microRNA. So in lots of projects, we simply want to study the coding RNA or just the messenger RNA. But in other projects, we might also want to explore non-coding RNAs, or even a combination of all the RNA, such as the whole transcriptome. So based on the target RNA you are interested in, we will offer dedicated protocols to sequence them and to perform the right analysis. So, but how can you know which RNA you should explore? So this brings on to the next question that you need to answer. So what do you want to discover about the RNA, such as profiling, quantification, or targets? Now, transcriptome is a screenshot of the gene expression profile in samples at a specific time point. So in order to choose the right RNA sequencing, I will suggest that you always start from your research objectives. So in some projects, you might just simply want to compare samples under different conditions or treatments. So you'll compare a treatment group and compare this against a control group. Or on the other hand, you may want to track gene expression profile across the time course of a treatment, such as a dose of medicine. Or you might even want to clarify differences within tissues with the same genome. So for example, the baby's eye cell and a skin cell. So why can the baby see the world with his big cute eyes, but not with his ear? So how is the gene expression regulated in different samples? So in order to understand their molecular differences, we will want to find out what transcripts or RNAs are being uniquely expressed, and also how many of each. And all of these questions can be more or less answered by using RNA sequencing. So in RNA sequencing, we basically have two major questions. So the annotation and quantification. We would like to know what RNAs are being expressed, or what are they. So however, you also need to bear one other thing in mind when studying messenger RNA, because splicing adds another level of complexity. As shown in eukaryotes, due to alternative splicing, a gene can produce different isoforms of a messenger RNA, which in turn gets translated to different proteins. So different exons are combined to produce slightly different messenger RNAs, and with the removal of introns. So therefore, if you are studying messenger RNA, you might want to consider sequencing the different splice variants as well. And of course, the second question is the quantification. That means we need to know how many of each RNA is present in your samples. But in some research, such as in the field of oncology, we are also interested in how gene expression is regulated. As shown in this figure, we have the microRNA, the long non-coding RNA, and circular RNA. And all of these non-coding RNAs can interact with different cellular molecules. And they interact in order to regulate gene expression. So, in non-coding RNA sequencing, we also want to know if there is any potential interactions between different types of RNAs, as well as the potential targets of this non-coding RNA. So now that we know what RNA you want to study and what questions you want to answer, finally, we also need to know what sequencing technology is available. How can you sequence the RNA of interest in your samples, and do this by using the latest sequencing technology available? So at Novogene, as a sequencing service provider, we have the most advanced platforms to cater to diverse sequencing needs. So we have the NovaSeq 6000 machines, and it's the most commonly used next-generation sequencer. On the other hand, Nanopore and PacBio are the most advanced long-read sequencers. So however, this can only be used to sequence messenger RNAs or RNAs with a poly A tail. NovaSeq can also be used to sequence all types of RNAs, so from the messenger to the non-coding RNAs. So these different platforms also deliver different read lengths. So NovaSeq will sequence up to 500 base pairs, while PacBio could cover almost 10 kilobases in RNA sequencing. Just a side note that if used for DNA sequencing, the Nanopore and PacBio platforms can sequence much longer reads. So in fact, as reported by Nanopore, Novogene made a record in DNA sequencing when we achieved an N50 of 18.2 kb. So some of you may wonder, why don't you just go for sequencing the long reads? After all, it makes sense if the reads are longer, the better it should be. However, this will depend because the long read sequencing has its pros and cons. By using this method, we will be able to sequence the full-length transcripts, and this method avoids the PCR amplification or fragmentation of the input RNA before sequencing. However, the long-read sequencers are not cost-effective and usually have high error rates, and it also requires extremely high sample requirement. Therefore, and also if you want to quantify your reads, you still need to use the NGS data. So in short, by delivering these full-length reads, it will perform very well to characterize the different mRNA isoforms. So this is also known as the isoform sequencing method or the iso-seq. So no assembly of short reads is needed, but remember, this method has its own limitations. So in summary for this section, how do you choose the right RNA sequencing? First, you need to know what is your target RNA, and then your research objectives. And finally, what platforms can you choose? So there's also just one more thing I would like to mention. You can also sequence multiple types of RNA in a single run, as indicated in the whole transcriptome sequencing service. That means in this service, we can deliver information for 2, 3, or even 4 types of RNA in one package. And if you are interested in this area, please feel free to talk to us. So now that you have thought about how and what RNA sequencing you're interested in, so now we move on to the next part, the service workflow. How can Novogene help you in your research project? So in this next part, I will go through our service workflow with you. So at Novogene, we provide end-to-end service for each project, starting with your samples and all the way to the bioinformatics analysis. So no matter your project, you can always talk to us and see how we can move the project forward. Importantly, we deliver high-quality data with publication-ready analysis. And our account managers and technical team are also on hand to provide a personalized service just for you. So for RNA sequencing workflow, no matter what RNA you are looking into, you can simply ship us your total RNA to start with. So once we receive your RNA samples, we will perform the library construction, sequencing, and bioinformatics analysis. And quality control checks are also performed at each step. So let's start at the very beginning. How can you prepare your RNA samples? So here I've just listed some common methods, and these are our recommendations. So the first is the Trizol method, where Trizol is a ready-to-use reagent and is designed to isolate high-quality total RNA from cell and tissue samples. And there are also a wide variety of commercially available RNA extraction kits, and which are normally quick and easy to use. So depending on your samples, you can purchase these kits and perform the extraction yourself. Additionally, we also provide an extraction service, but this is only on a project-by-project basis. So please drop us an email and we can help you as best as we can. Now after the extraction, and before you send out your samples, you should check how much RNA you have and how good they are, as different projects will have different sample requirements, and our account managers will happily provide the requirements for your project. So as an example, we would require a minimum of 0.4 micrograms of total RNA for an eukaryotic mRNA sequencing project. So here I've just listed some common QC methods that you can use. So an agarose gel can be run to check for potential contamination as well as degradation. You can also use a nanodrop reading, and you can use this to check your RNA amount and purity. And you can also use an Agilent machine to check the RNA integrity number, and it usually ranges between 1 to 10. And again, the higher the RIN, the better the quality of your samples. And on the bottom right is just an example of an Agilent bioanalyzer output and showing its RIN, number, concentration, and other information. So note that once we receive your samples, we will also run a QC check. And in fact, we will use all 3 methods to check your sample quality and quantity. And we will provide you with this report, and you can decide to either proceed to the next step, or you can decide to send us replacement samples. Now you might ask, how do you send your samples? So for RNA samples, we recommend dry ice packaging. So please put enough dry ice in the packaging, as normally 5 kilograms of dry ice is consumed per day. You can ship using any courier of your choice, such as FedEx or DHL. Our account managers are also ready to help with sorting out your shipment order. And we will provide a sample shipment checklist. And basically, you only need to follow the instructions and inform us of your tracking number when it is available. And while there are additional issues since the UK has left the EU, we can provide guidance for shipping from EU to the UK. So unfortunately, not all of us can prepare enough RNA for library preparation, but don't worry. If you only have limited RNA, we do have a method for ultra-low input amounts. We have the SmartSeq ultra-low input RNA kit available, and this is mainly used for single-cell RNA or ultra-low amounts of RNA. This kit is designed to generate high-quality, full-length complementary DNA by cDNA synthesis directly from 1 to 1,000 intact cells, or 10 picograms to 10 nanograms of normal RNA. And based on our experience, this protocol works very well for RNA amounts of more than 1 nanogram or around 100 cells. However, this method also has some disadvantages. This option is only for eukaryotic messenger RNA sequencing or RNA with poly-A tails. And samples after this amplification cannot be analyzed together with the regular or low input samples. And just to show you as an example of this method, on the left, as you can see, this sample only has 4 nanograms of total RNA. And after using the SmartSeq kit, we obtain much more RNA material as well as higher molecular weight RNAs. So by using this smarter amplification method, now we have enough RNA for library preparation. So after checking the RNA is of good quality and quantity, the next step is of course the library construction. Here, I'm just using the library preparation for messenger RNA sequencing as an example, the poly-A enriched and non-directional library. So to start, the mRNA is firstly enriched for all poly-A transcripts from your sample. The mRNA is then fragmented into different lengths. This then undergoes the first round of reverse transcription, and the RNA template is then removed, and second strand cDNA synthesis is performed. Next, the ends are repaired with the addition of A's and the ligation of adapters. Finally, this then undergoes a few rounds of PCR amplification. So you will end up with a product containing adapters and a DNA insert. So this method of poly-A enrichment of mRNA results in the construction of a 250 to 300 base pair insert cDNA library, and this is now ready for sequencing. So poly-A enrichment is the most commonly used method for mRNA-Seq, as well as all messenger RNAs have a poly-A tail in eukaryotes. So however, for other types of RNA or highly degraded RNA, such as RNA extracted from FFPE samples, poly-A enrichment is no longer efficient. So we will use different library preparation methods, such as the ribosomal RNA removal library. So if you remember the pie chart I showed earlier, ribosomal RNA accounts for almost 90% of total RNA. So for non-coding RNA, such as long non-coding, we usually use a ribosomal RNA removal to enrich them. So while for small RNA, we will perform a size selection to enrich for this group of RNA. And just to highlight, if you prefer to construct the library by yourself, that is fine, as we are happy to receive that as well. So there is also the PacBio and Nanopore libraries for the long-read sequencing. And I would like to elaborate on the whole transcriptome sequencing at this point. So in this case, we will prepare two libraries using the total RNA provided. So one is the ribosomal RNA removal and directional library to target the long non-coding, messenger, and circular RNA. And the other is the small RNA library to target the small RNAs. So this allows you to not only explore the RNA profiles and the target prediction, but also dive into the network to study, for example, how the messenger, circular, long non-coding, and microRNA interact with each other and their involvement in the regulatory network. But if you check our sample requirements, you will only need 5 micrograms for this project. And this is half of the total RNA requirement needed for running each RNA type individually. So this now brings me to the next stage of the workflow, sequencing. So how long are the sequencing reads obtained? So the abbreviation SE and PE refers to either single end or paired end. And in the single end, only 1 sequencing read is obtained, whereas in paired end, 2 sequencing reads are generated from each library molecule, so 1 from each end. And the number following SE or PE refers to the length of each read. So this can either be 50, 150, or 250 bases for Illumina platforms. And for each library type, we recommend these strategies, and you can see them on the table. So for messenger, long-non coding, and circular, we recommend the paired end 150 strategy, while for small RNA, it is single end 50. And for pre-made libraries, you can customize this as listed here. So just to summarize this part of the talk, only total RNA is needed for us to initiate your workflow. And if you have low amounts of RNA, we can use the ultra-low input RNA amplification method to increase the sample amount. We can also provide different library preparation methods to target each different RNA types. And multiple sequencing strategies are also available. And we will move on into the bioinformatics analysis for the next 2 sections. But before I move forward, I also want to inform you of our fast mRNA sequencing service available in the UK lab. So that is the Novogene lab in the Cambridge Science Park, and we can perform the RNA-QC, automated library preparation, and sequencing. And we are able to provide a fast turnaround time, and this can be as short as 2 weeks. So please contact us if you are interested in using this offer. So now I've covered your RNA samples and the construction of the libraries and the sequencing. And now it's time for us to start your bioinformatics analysis. So here is just a general messenger RNA analysis pipeline. And we start from the raw data, and then we move on to the analysis step. This might seem very complicated and confusing with all the different terminologies, but just take a breath and remember your research objectives. So if you still recall your research objectives, we are trying to answer 2 questions. So first, we want to quantify your RNA samples, your RNAs in the samples. And the second, we would like to characterize them by checking for structural analysis, such as alternative splicing and mutations, such as single nucleotide polymorphism, SNPs, or insertions or deletions, indels. So actually, if you're interested in all the details, we have several on-demand webinars to go through the detailed analysis for messenger RNA and non-coding RNAs. And we actually have more planned in the future. So if you need the bioinformatics support from Novogene, we will provide all of this information based on your experimental design. So however, it just takes a few simple steps for you to design your analysis with us, and you can do this by simply filling out our bioinformatics form. So we will provide you with a bioinformatics form to fill, and you will just simply need to fill in the form, and we will run the analysis for you. So firstly, you will need to name your samples and groups. So the sample names will be shown in your report, and results package as indicated by the arrows. So group names will let us know if any samples are from the same or if they're from different groups. And we can then use this to compare their RNA abundance between groups or use them for various other analysis. Step 2 is to choose the reference genome. And you can use our database, and it covers a whole range of different species, from mouse and human to less studied species like mosquitoes or even the elephant genome. And all of this is available in our Novogene database. You can also go to the appropriate browsers such as NCBI, UCSC, or Ensemble, and use those reference genome by filling in the table with the correct download links. Or if you would like to provide the reference genome that you have assembled yourself, you can also do this and provide us with that information. So thirdly, you also need to design your comparisons. So for example, if you would like to compare treatment versus control, you will just need to fill in the group name from part 1 here. Then we will know which is the treatment and which is the control group. So something to remember is if you flip those two around, then the result should be interpreted in a different way. So I'm just showing here as an example. The volcano plot is a very commonly used figure to present RNA sequencing results. And in this figure, significantly up and down regulated genes are shown in red and green, respectively. And in this experiment that I'm showing, we are comparing 6-hour group versus a CK group. And we have 5,000 upregulated genes and 4,900 downregulated genes. And if we change the comparison to CK versus 6 hours, then the results and numbers will possibly be very different. Another table you also need to fill in is for the Venn diagrams. So this is for us to check for unique as well as commonly differentially expressed genes among the groups. So this can be set up with 2 to a maximum of 5 different groups. So that's it. So just to summarize, if you need our bioinformatics support, all you need to do is fill out our BI form. And this is just 3 easy steps. So step 1, name the samples and groups. Step 2, select your reference genome. And step 3 is to design your comparisons. Again, here I'm just showing you the messenger RNA service pipeline as an example. But for other services, it is a very similar process. So now that we have looked at setting up your analysis, let's check out how your results will look like. And we are back to this pipeline again, and I'm still using the messenger RNA analysis pipeline. So basically, we will include the raw data and data quality checks to check the data quality, and then you will have the mapping results, which is in this mapping to reference genome section. And then this is the analysis section. So here's the quantifying gene abundance, followed by differential expression analysis and functional analysis. And the other analysis is the structural analysis, which is the alternative splicing, SNPs, and indels. So at this point, if you do not need bioinformatics analysis, we will only deliver the raw data with its data QC report. If you choose to include the bioinformatics analysis, we will deliver publication-ready results for each individual steps. So let's have a look at some of the results. So the raw data generated by Illumina sequencers are in the FASTQ format. And in FASTQ, each read is represented in 4 lines. And line 1 begins with the @ symbol and followed by sequence identifiers and optional descriptions. Line 2 is the sequencing read, and this is the actual sequence of your data. Line 3 begins with a plus sign, and this is optionally followed by the same Illumina sequence identifiers and description as in line 1. Now in line 4, this is the data quality values for each base, and this corresponds to the data in line 2. And if you use a parent sequencing, you will receive 2 FASTQ files for each sample, as shown here. But if you choose to use single-line sequencing, you will only receive 1 FASTQ file. So in the data QC, in the raw data QC, as these figures indicate, we will generate the distribution of the read error rate, the read quality, as well as the base percentage across the 2 reads. And all of these will help us to check how good the data is. And Q30 is a commonly used parameter for data quality, and generally, again, the higher it is, the better. Illumina officially guarantees 75%, while at Novogene, we will guarantee 80%, and that means more than 80% of your total data are with extremely good quality. For the other steps, we will also deliver the results in a table in Excel format or publication-ready high-resolution figures. So for example, we included all detailed information for mapping, like the total reads, as well as the total reads that are mapped to the reference genome for all of your samples. We will also include a pie chart to show the distribution of mapped reads across different genomic regions. And this will show the percentage of reads that maps to either exons, introns, or to intergenic regions. And the graph on the right shows the distribution of map reads on chromosomes. So the outer ring is the chromosome number, the middle is the chromosome band type, and the inner ring is the distribution of all the reads. And these are just common methods to show your results. And you can also contact us to customize the figures if you prefer. Now, the mapping results can also be visualized using the Integrative Genomics Viewer, or IGV for short. This is a common software, and we use this to visualize mapping results to see abundance of reads across the genomic regions. And IGV is a freely available software and is open source. You can download your reference genome and map results in band format for your samples into the software, and it will display it for you. Of course, you can also change the setup to also show read abundance or annotations, among other things. And in your results package, all delivered files are in the proper format, and these are all ready to use with IGV. Now, I touched on this earlier, and volcano plots are used to infer the overall distribution of differentially expressed genes. Now, this is a type of scatter plot that shows statistical significance versus the magnitude of change. And I assume you still remember the BI form that you have to fill in to compare group A versus group B. And each point on the plot represents one gene. And in the volcano plot, the x-axis shows the log-fold change between A versus B. This means that positive and negative values indicate high and low abundance respectively. And this is detected in group A when compared to group B. The y-axis shows the p-adjust value, or the statistical significance threshold. Here, it is indicating our cutoff for significance, and that is the p-adjust is 0.05. So everything below is not significant, while everything above is statistically significant. So basically, in this example, we identified this number of significantly upregulated genes, shown here in red, while the green points show the significantly downregulated genes. And again, if you change the comparison to B versus A, then the result will be very different. So in summary, by using volcano plots, you can get an overview of all detected genes with their abundance in different samples or groups. Another result that we provide is a heat map. This is also commonly used, but only focuses on differentially expressed genes. So in this plot, all differentially expressed genes with similar profiles are clustered together. Each line indicates one gene. As shown here, red indicates upregulation, while blue indicates downregulation. And we can also obtain more gene information, such as the chromosome number, the transcript type, and also the transcript length. An advantage of heat maps is gene function prediction, and we can predict gene function prediction by performing data clustering. So by clustering the genes with similar expression patterns, it is possible to predict unknown functions of previously characterized genes or unknown genes. So due to the time constraint, I cannot show you all details of our result package. However, we will provide a directory for you to easily navigate through your data. So this is the mRNA sequencing data directory as an example. And so you will be provided with two FASTQ files because, again, this is pair-end sequencing. The BAM files are also provided for mapping, and together with the reference FASTA file, with the reference genome FASTA files, you can input this into IGV for visualization. And of course, the IGV manual is also included. And you will also find the project report, results, and instructions. Now, MD5 is a tool to check your data file size in case of data download errors. So we also include the Novo Smart tool, and this is for customizing figures. And you can also use this to change the parameters to adjust your figures. But this is only included in the RNA sequencing with a reference genome project. So again, this is the pipeline and the result directory side by side. And if you compare the pipeline and our result package, it actually corresponds to different parts in the workflow. So if you're interested in the complete pipeline and other pipelines for other services, please feel free to contact us. And so with all your publication-ready data, it is now time for you to publish your amazing work. So in our project report, we also include a reference as well as a list of software used. So you can find the information for your software from the software, the name of the software, the version, the different parameters, as well as any remarks in your report. So if you need our support for your publication, we are definitely happy to help. So I hope that now you have a better idea of how to carry out RNA sequencing. I hope that you can now choose the right RNA sequencing service based on your research objectives. And at Novogene, we provide end-to-end service for each project, starting with your samples and going all the way to bioinformatics analysis. And we will work together with you to design your bioinformatics analysis. And this can be done in the 3 simple steps. And we will deliver high quality and publication-ready results. And just to quickly summarize about Novogene, we also provide other services, such as for DNA sequencing, epigenetic services, and customized services. So please feel free to talk to us if you're interested. And hopefully, we can be involved in your next publication. And with that, we are at the end of my presentation. And thank you very much for listening. And here's a list of our local Novogene account managers covering Europe. And please do get in contact with them. So I will now pass the session back to Shama for the Q&A session. Thank you.
Right, I've remembered to unmute myself this time. So we've had a lot of questions, so we're going to try and get through this in the time that we've been allocated. I'm going to now obviously move on to questions and answers. I'm going to now bring in Wee, who is our product manager, who will help bring in some additional expertise to this part. Many of you might already know her from previous webinars. So we've got quite a few questions. I've got it on my phone. So I'm going to start off with a question for Wee. It's from Christos Clement, and it says, can FFPE cells be used to study expression? I'm interested in analyzing the whole transcriptome.
Thanks for the question first. Actually, for FFP samples, the big issue will be the degradation of the RNA input. So we do offer the library preparation by using the ribosome removal method to enrich those RNAs. So if you are asking about the whole transcriptome, referring to cover all types of RNAs, such as messenger RNA and all types of non-coding RNAs, we have to make the decision based on the data, but usually we do not recommend you to do that because the RNA are usually too degraded. And with those very small fragments, it doesn't make too much sense to work on the structure related or the non-coding RNA studies. So we usually recommend you to perform the messenger RNA quantification. So for the FFP samples, that should work very well.
Great, thank you. I have another question from Lizo, and this one's for Joseph. It says, do you also supply meta-transcriptomic services?
Yes, we do have a meta-transcriptomic service. And again, so please do get in contact with our account managers, and they can give you, customized and tailored response to your project. So if you let them know, what you're interested in, give us a bit of more details, and we can give you more information about the services that we provide. So yeah, so please do get in touch, and yeah, we can definitely perform the meta-transcriptomic service. Thanks.
Yeah, I've got another one for you, Joseph, and this one's from Kirsten Westendorf, and she says, how do you know how much gigabyte to order for different sample types?
Well, this will generally depend on what kind of organisms are you, what's your species of interest. So you will need to know, for example, what's the genome size and how much coverage you will want for your data. So this depends on quite a lot of factors that we need to figure out. So so this will, like I said, the genome size will help, as well as the coverage that we will want. So again, get in touch with us about the species. So that's a very key information that we might need to think about in order to figure out how much gigabase of data you would like.
I can add more comments on this.
Yeah.
So actually, yeah, you are right. So it really depends on the species information, because the transcriptome and the genome size are greatly different across species. But basically for the eukaryotics, in previous research, so 6 gigabase of the data will cover the messenger RNA very well, because they have very high abundance in the transcriptome. However, for some other types of RNA, such as the long non-coding RNA, the circRNA, or small RNA, they are expressing with relatively low abundance. So for those RNAs, we probably need to sequence more data. Like for long non-coding RNA, we usually recommend them to sequence between 9 to 15 gigabase. So it really, we make the recommendation really depends on the type of RNA and also our previous project experiences. So if you give us your species information and what type of RNA you would like to sequence, we can offer better recommendation for that.
Great, thank you. We have another question for you, Joseph, and it's actually one of our own clients, Sunal. Hello, Sunal. She asks, will raw counts be provided to us after bioinformatic analysis?
Thanks, Sunal, for joining us. So like I mentioned, we will provide the raw data, and so you will also have the raw data quality checks as well. And so we'll perform the bioinformatics analysis, and all the results are provided to you. So the BAM files, the reference genome files, and all of this is included in our services.
Great. Another one for you, Joseph. This one's from Lisa Nolan, and she says, is there a limit on the number of comparisons we can ask you to run?
So we do have a standardized pipeline. We will do all these steps that I've mentioned. So if you want detailed information on them, so please do drop us an email. But if you are interested in what comparisons or different types of analysis, we can look into that as well. So if you just give us a bit more information, such as what kind of software or what kind of comparisons that you want to do, we can definitely give you a more detailed response and a customized response for your individual projects.
Great. I've got one for we here. Bacha Chiab asked, is really PacBio interesting for RNA sequencing? What is the average length of splice variants?
Well, PacBio, usually we can prepare the library for 10K insert size, 20K or 30 or even 40K. So they can sequence quite long compared to the NGS sequencers. However, for the transcripts, especially because this could be used for sequence the messenger RNA in the eukaryotics. For those transcripts, they usually range between like several hundred base pair to 10 KB base pairs. So when we prepare the libraries, we do see a peak at around 2 KB or even 4 KB. So by using our pipeline or using our library preparation, we do have options. So if your sample are quite perfect, like with the ring number very high, it's possible for us to capture those peaks, such as around 2 KB. Or if you really want to focus on the short fragments or short transcripts, So we can also perform the set selection. So such as we pick up the fragments, we pick up the transcripts lower than 2 kb, or even we can perform the site selection by choosing the large pieces. So we can perform the site selection and focus on the transcript longer than 2 kb only, or you can mix and match. So it's possible for us to capture or focus on different range of your transcripts.
Thank you. I've got another one here for you, Yi. She's actually asked 2 questions. So the first is from Magdalena Yaris, and she asks, what kind of not so advanced user-friendly programs do you recommend for RNA-seq data visualization?
It really depends on which step we are talking about. So for if you are processing the raw data, so there are a lot of entry-level programs you only need to, or applications you only need to upload your data, such as the faster QC. So you can perform the data quality check by uploading your raw data, and it will deliver the quality distribution for your data. Well, for the further step, like the mapping, as Joseph mentioned in his presentation, we can use the IGV software. So you can load your reference genome and your mapping result. And with that, you can check the abundance of your reads or across the genome regions. And you can also check if there is any, like the breakpoint between your reads, because sometimes the transcripts may have some alternative splicing or even fusion between different genes or transcripts. But for the further steps, such as the differentially expressed genes, as Joseph introduced, the most commonly used method will be the volcano plot and the heat map. So for that, you have to use the R program to produce those figures, and they have different package to produce those figures. Yeah.
Great. Magdalena also asks, and this is to you, Yi, is it possible to visualize RNA sequencing data for publication purposes using only identified differentiationally expressed genes and values of down and upregulation of these genes? Experiment is about to compare mutants of Arabidosis to wild type.
Yeah, that's quite possible. So again, so I think for your purpose, the heat map will be better for the visualization. So you can only focus on the differentially expressed genes. And if you want to customize how you want to visualize it, we can simply add like the values, the up regulation abundance or down regulation abundance on those small squares. So because in each square that will presenting your gene in each sample. So with that, it will help us visualize more directly to check their abundance.
Great, I've got one here for Joseph, and it's from Demetrius, and asks, how long does it take, roughly, to receive the bioinformatics results since you receive the samples?
So, yeah, it will largely depend also on the number of samples that you run. So, if you have one sample, we could, do it within, with our fastest turnaround time service as well. We can go as quickly as, 2 weeks. But you have more samples, then we will need to check, how long this will take. And normally it will range between, up to about 20 to 25 days. That's on average. But I would like to stress that this will depend on the number of samples and what kind of analysis that you want.
Great, thank you. I've got one for Wee, from Apuva, sorry if I pronounced that wrong. Do you deplete rRNA and globin before library construction? I recently had RNA sequencing performed on RNA isolated from blood. Though the kit said that the rRNA and globin will be depleted, we had over 60% reads mapped to rRNA and globin. How much effect does such contamination have on RNA sequencing?
Well, it's very common to perform the ribosome RNA depletion and the globin messenger RNA depletion for blood samples. So, but the ribosome RNA ratio is really depends on a lot of factors, such as how the kit performed during the depletion, like their depletion efficiency. And also it depends on your sample quality as well, such as for the very good RNA, so it's quite possible we get lower or we get very little ribosome RNA after the depletion. But for some highly degraded samples, it's quite possible because the fragments are too small, and it's possible for them to have a little bit to have high ribosome RNA ratio. So it really depends on a lot of factors, but we cannot make the conclusion. So how well the depletion will be for all samples.
Great, thank you. I have another one for from Nicole van Dienen. Are the provided volcano plots directly linked to the raw data so that I can see which sequences are up and down regulated?
So you mean the live volcano plot? Yes, so for that one, we do process the request, and it is possible for us to provide the live volcano plot. So by using your mouse to click on or to move close to each point, it's possible for us to show the gene information or the up number or the down regulation number. So it's quite possible for us to provide that. However, for that, it will be shown in like the web page. So this file will be super big because it has stored a lot of information in it. And I'm not quite sure if this is quite good for you to have the publication, but you can attach it as a supplementary information as well. So if you do need this type of volcano plot, feel free to contact us. We definitely can do that.
Great, we have so many questions and we'd love to go through all of them, but we're going to give you one more last question and then I'll just wrap up. So this one's to it's from Tamara. How do you control you have a good representation of your transcriptome in your library when you consider a library has a good quality to work with? So I think it's more to do with construction of libraries. And how do we know that we're going to get good constructions of libraries?
Yes, so as Joseph introduced, we do have the quality check before each step, such as when we receive the samples or after the library construction. And for the different library preparation protocols, we try to dedicate each method to target our target RNA, such as for messenger RNA, we use poly-A enrichment. So this is quite efficient for us to capture all the messenger RNAs. Well, for the circRNA, after you perform the ribosome removal and the following steps, we also remove the linear RNA to enrich the circRNA. So for each library, each type of RNA, we do dedicate a specific protocol to try our best to enrich all of them. So after the library construction, we do perform the library QC to check if we have a good distribution across the peaks, and if we got the target fragment size as we need. So I think that will help us to deliver the good library for sequencing.
Great, thank you. Like I said, we have so many questions and unfortunately we can't answer all of them, but we will get back to you. It might take you a couple of days, but one of the members of the team will get back to you guys. So that's it for today. I'd like to thank you all for taking your time today to spend with us. We hope you have enjoyed the webinar and found it useful. We will send you an email with the link to the webinar recording, because I know a lot of you have asked for the recording, and a slide deck. This will also include a very short survey, and we would really appreciate it if you could take the time to complete the survey so we can improve on our webinars for the future, and also to make sure it's relevant to you guys for your research goals. You can expect to receive the email from us on Friday. In the meantime, please stay in touch, and you can follow us on Twitter, LinkedIn, or you can get in touch with us via the website at novagene.com, and we look forward to, especially myself, hearing from you soon. So thank you very much, guys.
Thank you all.